So let's now turn to the second keynote, which is by Markus Heinrichs. He's a professor of biological and personality psychology in Freiburg, in Germany. We have known each other for a while. Yeah. And he is specialized in oxytocin research and is actually one of the pioneers in looking at the overlaps or the associations between oxytocin and social behavior and social cognition. And you're going to talk about today what all this means for us working with autism spectrum disorders. Yeah. And when I listened to you the first time, Markus was also lecturing about the Trier stress test, which is actually often used also in oxytocin research. And I hope you will tell us something about this today also.
I will.
That's good.
Okay.
Welcome to Stockholm.
Thank you. Yeah, thank you, Sven, and good afternoon, everybody. First, I would like to formally thank also Sven Bölte, Monika, and the others in your team for putting this conference together, which is also an exciting conference, a very exciting conference for me, and especially here in this beautiful city of Stockholm. When I agreed to come to this conference 2 years ago, I was very optimistic that I can bring tell you some, let's say, initial clinical evidence for oxytocin in autism, maybe also some first clinical recommendations to treat individuals with ASD with oxytocin. But I have to say it's unfortunately too early to do that. Because all studies that deserve the term clinical trial, serious clinical trial, large-scale clinical trial, are currently running or are just in a planning stage or are starting now worldwide in many, many places. So I could easily stop my talk, of course, now after 60 seconds, save some time for the others. But no, I think seriously that I'd like to invite you to come with me to a journey through the knowledge, to the really evidence that we have on oxytocin in humans, related to behavior, related to, of course, social behavior. I, of course, show you some preclinical data with different, let's say, social disorders. We also have to tap into genetics a little bit to understand something about the sensitivity of the receptor for oxytocin. And of course, we should have some time left, hopefully, to discuss about the chances that we still face and about the limits that we should also know about when we talk about oxytocin. Because oxytocin is in the media, it's fashion, there is no psychiatry hospital at the moment in the world, as I know, where it's not running an oxytocin study. This is not necessarily helpful for this kind of research, but I think I will make some comments on that today. And the good news is that there is also no study telling us that oxytocin does not work in social disorders, including autism. So I think there's not only hope, there's also evidence how to work if we proceed with clinical samples. And that's, I think, a good reason to give this talk now, the next 45 or 50 minutes. It all began, I should say, in the 90s, in a time where we had so much literature from epidemiology, from psychiatry, from cancer research, from research after heart attack. And all this research on somatic, mental, psychosomatic disorders showed that positive social interaction, especially social support, exerts these powerful, unspecific positive effects, protective effects on health. And the question was always, I think, what is behind this? What is the difference between the two brains, two immune systems, two autonomic nervous systems, two, I don't know, other biological systems? If one individual feels attached to others, feels in a secure, stable social network, and the other individual does not perceive that or does not have that. And we all know that having these stable networks, being able to socially interact, is a very strong factor, positive protective factor. And then there came data from animal research in the 90s especially, I think also in the newspapers, and I have to torture you with 2 slides on animals, and then I will, of course, start with human research. But this is fundamental to understand, because without this kind of research, we would never have started in my lab in the 90s with oxytocin in humans. And it was not really cool in these first 5 or 6 years because nobody was interested in human research. But the animal research just showed, as I think, important messages. These are brain slices through the brains of prairie voles, and this is a montane vole, and these species differ regarding their attachment behavior. So a prairie vole, normally in the wild, but also in the cage in the laboratory, would sit next to the partner in physical contact, in one corner of the cage. They show very extreme kind of monogamous pair bonding behavior after mating. They show paternal behavior, so fathers and mothers take care of the youngs equally. And the montane vole looks the same, but shows the opposite in terms of behavior. Montane voles live alone, they mate and separate, and they show no paternal behavior at all. If you have a look to the brains, you see these dark areas. These are just labeled. This gives you a picture of the density of oxytocin receptors in the brain, and the highest density here in the socially living prairie vole is in the nucleus accumbens, which is important for reward processing, and in prelimbic cortex areas. So these are all areas relevant for reward processing, especially when it comes to social behavior. And we know that the montane vole has virtually no receptor for oxytocin in the accumbens at all. Now you can do some nice and elegant behavioral studies with these animals. For example, the lady, prairie vole lady here, or montane vole lady, is alone in this cage, and she can visit her partner with whom she was mated before, or a stranger male in the yellow chamber. As you see, montane voles prefer to stay alone and equally visit these 2 chambers, and the prairie vole prefers to stay and spend much more time in the cage of her husband, so to speak. What you can do now is you can block the oxytocin by giving an oxytocin antagonist. That means oxytocin cannot exert its effects anymore. And if you do that in prairie voles, you block oxytocin receptors in the nucleus accumbens. What you see here in terms of minutes in the cage, these prairie vole ladies would reduce dramatically the time they spend with their husbands, and they have much more interest now in the stranger male in the yellow chamber. So you can really switch on or off monogamous pair bonding behavior, in this case in mammals, with one neuropeptide. That was fascinating when I read that in mid of the 90s. And there was another interesting story. And that story is that in many mammals we see that in the amygdala, which is an interesting and important region for not only for fear and anxiety processing, but also for alertness, for an alarm response of the brain. In this amygdala, you see this high density. This is in rats, research in rats, high density of oxytocin receptors here in this central amygdala region, surrounded by receptors for vasopressin. And this is important because if you stimulate these receptors here in the amygdala can then shut down, or oxytocin would shut down the input into the periphery, into the brain stem, and that means that the fear response, the autonomic fear response, is better controlled at the level of the amygdala via the oxytocin system. That's also important if you think about oxytocin in humans, because we have these 2 stories, to make it more simple now. On the one hand, you have this pro-social rewarding thing, that social interaction is obviously more rewarding. This is the first part I showed you. And then we know that oxytocin reduces, let's say, stress-associated, stress-related, fear-related systems like the amygdala or like the hypothalamus pituitary adrenal system or axis, and also reduces, of course, cortisol. This is not only true for, as we know in the meantime, for mammals. We also know that there are homologs, so very similar molecules in birds, in frogs, in fishes, and it goes down to worms. So also sexual behavior in worms is different when you block this oxytocin vasopressin homolog in worms. So obviously a very old and very relevant molecule for some species conserving behaviors. So people told me that sounds like the brave and pro-social tiger mouse animal that looks like this. That is, of course, not existing in reality. But the important question for us today is, first, how to translate the knowledge that we have from animal research to humans. That has been done the last 10 years. And from so-called healthy humans, from neurotypical humans, to patient populations where we think they have problems in their social behavior and or in controlling of social stress in different situations. And before I start with data, I would like to give you some basic information about the knowledge that we have about the paradigms we use in this kind of research with healthy controls, but also with patients. The first important thing is that, of course, we produce, as voles, as mice, as rats, we produce oxytocin in the same structures in the hypothalamus, in the brain, and it is then processed along this axonal projection to the pituitary, the posterior pituitary. From there, it is released to the bloodstream. That's what medical students learn in their textbooks since the 50s, 1950s, that you need oxytocin for breastfeeding, you need oxytocin for parturition, and this is the medical function, if you like, the physiological function. But for us today, of course, it's not so relevant to have a look on the periphery. We are interested in the brain today, in behavior. So what is going on in the brain? What is oxytocin doing there? We know in these days that oxytocin is not only released from these terminals, axonal terminals here, but it's also released from dendrites. And this dendritic release makes it possible that diffusion brings oxytocin not only to close regional structures here, but also to distant targets, specifically in the limbic system. Show you brain imaging data also later. It's also important to know, before I show you behavioral data, that we have at the moment one tool where we can, in the meantime, say, without knowing exactly the pathway, but we can say that intranasal administration of neuropeptides, not only vasopressin or oxytocin, but also many other, like insulin, can reach the brain after intranasal administration. So this is increase, as you see, these are vasopressin, in this case, levels in the cerebrospinal fluid after lumbar puncture. Now you take brain liquor, and you can see that very slowly you have this nice increase, which is of course not visible if you administer placebo or if you administer neuropeptides intravenously. So the intranasal, the nose-brain pathway seem to work at least for 1 hour, maybe 2 hours, to increase levels in the brain. We also have a better picture in the last years about the receptor. This is important because when we think about how to use also changed molecules, more specific molecules, we have to have a good picture of the receptor in the brain, which is the same like in the periphery, and also about the cascade of messages in the cell, which can be different, of course, from cell to cell, and is also an important field in research. Very recently, we could show, this is unpublished data, that in humans, this is post-mortem brain tissue analysis, and you see that these brown spots here are just from immunostaining studies, immunoreactive cells, oxytocin receptors. And what you see is that we have a really high, the highest density in humans in the nucleus accumbens, which is not trivial. We were hoping for that, but it's exactly like in the prairie vole. And the second structure where we have so many receptors is the amygdala. We know that between 60 and 72% of all neurons in these two areas contain oxytocin receptor neurons, which is relevant when we think about working with oxytocin in patients. We also know that if you show subjects in the scanner, in the magnetic resonance imaging, pictures from faces, social information, then it doesn't matter whether this information is a positive or negative one. If there's a social information, then people in the oxytocin group, this is the gray bar here, clearly showed lower amygdala reactivity as compared to the white bars, the placebo group. This is also, if you want, a very old and very well-known biological mechanism that higher levels of oxytocin make it more likely that you show social approach behavior and that you can overcome avoidance behavior. And that means you have to keep control of the amygdala. You can easily show that by using neuropharmacology approaches like oxytocin intranasal spray. We also know that oxytocin increases trust. I show you an experiment on that later. But just to mention the brain again, because I don't have the time to go further into the brain in my talk, just to mention that here, we know that this increase of trust under oxytocin is explainable from the pictures of the brain by a better control of, let's say, fear responsive systems. It's not only the amygdala, it's also the midbrain. It's connectivity between amygdala and midbrain, and it's also the caudate nucleus, which is relevant here and is also relevant for reward learning. Looks complicated, as Chris also showed pictures like this, but the big difference is here, I'm not talking about syndromes, I'm not talking about deterministic things like genetic diseases. I'm talking here about variants, common variants in our population. And there's one that seems to be really relevant for our topic today, which is RS53576. This is a variant where we know that 15% of the normal population have a so-called risk allele. I'm not happy with this. It sounds very deterministic, but these 15% are different when it comes to social behavior, empathy, stress responsiveness. I'll show you that later. So come back to that in a minute, but also this kind of research is relevant because we would not like to give oxytocin to all patients with social phobia, to all people with autism. I think we should not make the mistake that psychiatry is doing for so many decades to give a substance to all depressive patients and then have a look what happens. Maybe we can make it in a little bit, can make it better by following a pharmacogenetic approach and identifying subgroups that might really benefit from oxytocin substitution. Because these SNPs, especially the one I mentioned, are associated with functional and structural changes in the brain in those areas which process social-emotional behavior. So there's a clear association, and it's shown several times now, that these specific brain changes are associated with these variants in the oxytocin receptor gene. So to sum up this from a methodological view, of course, we all started with behavioral testing, trust games, the stress test Sven Bölte mentioned. Then we found out we should know what happens in the brain, especially using fMRI. in recent days, to have a closer picture of all that, we use brain tissue analysis to understand the receptor distribution in the brain. And to manipulate the oxytocin levels in the brain, you need neuropharmacological approaches, applicating oxytocin or other drugs, and also measuring hormones. And as I said, we're also including more and more approaches from neurogenetics before we go back to the beginning and do that, what we like to do when we started 15 years ago. I think now time is ripe to concretely do translational medicine research. That means combine, for example, neuropeptide administration with specific, more behavior-oriented therapy to really change then social behavior. So, I mean, it's clear that this is always at least the wish to follow this biopsychosocial approach. I'd like to give you 3 examples now how we did that and how we're still doing that. The first study was not a nasal spray study because we were not brave enough to administer it in patients or in males. So what we did is we tried to use the Mother Nature paradigm, which is the lactating or the breastfeeding woman. And what we did is we invited women 8 weeks after birth to the laboratory, and they came with a baby and with an accompanying person. They breastfed the baby or they just hold the baby in this 15-minute period. So we just manipulated or varied the distance from breastfeeding to the stress test. One group breastfeeding here, the other group was breastfeeding here. And then babies were handed over to the accompanying person, and then all women underwent the Trier social stress test. The Trier social stress test is not a very nice experience if you undergo this. What you do here is, this is the standard in human research if you like to socially or psychologically stress people in a serious way, but that the ethical committees still happy with that worldwide. So it's done in 10,000s of subjects, patients, and healthy people. What you do is, it's like an application interview, like an interview committee, a panel of evaluators. They are not mean, but they are looking in a way which I would call neutral. So they are not very supportive, they are not smiling at you, they are neutral, they are staring at you, they write something down, and you have 5 minutes of presenting yourself. And most people stop earlier. Then these people would tell you, you have 2 minutes more, please go ahead. If you can't go ahead, they will have some specific questions. If this is over after 5 minutes, you have to count backwards, which is not said before, from, let's say, 2023 in 17 steps, which is also not so relaxing for most people. What you normally also do is you measure cortisol in saliva or in the blood, you measure epinephrine, norepinephrine, heart rate, blood pressure, and of course subjective ratings. So you have a nice picture of the stress responsiveness, and believe me that stress responses had showed 2 to 3-fold increases in these parameters. What you see in this lactating study was that in women just holding the baby, you see it very normal. This is plasma cortisol, this is saliva cortisol. You see a normal cortisol stress response over time. This is the 10-minute of stress. And those who breastfed her baby 15 minutes before, they have for 60 minutes a very nice reduced cortisol stress response. That is a very old and in all mammals existing protective mechanism for both. The focusing of the mother on the baby is only possible if your alarm systems are under control. And the baby is protected also against, for example, glucocorticoids in the milk. So this is also for attachment research, a very basic biological mechanism that is also, of course, existing in humans. Physical contact is another endogenous stimulation. You don't always need intranasal spray. This is a method that works in animals. You can stimulate bellies of rats and mice, and they show oxytocin increase. We also knew from research done by Becky Turner in San Francisco that oxytocin goes up after a massage, a neck-shoulder massage by a physiotherapist. And this is not the case if you see your favorite movie or listen to your favorite music. And we thought this is interesting to combine that with stress, social stress in the laboratory. So we invited women who should come alone. This was, of course, randomly assigned. The second group was asked to bring their partner or their husbands with them, and the husbands were explained or instructed to verbally support. You can say everything that you want to say, and that helps your wife to reduce stress. And the third group received from the husband physical contact, and that was standardized, of course. So husbands were shown how to administer a neck-shoulder massage, and they were allowed to do that, but they were not allowed to talk. So that was a verbal, and this is a non-verbal, if you like, couple interaction. And then, of course, males, all men left the room, left the laboratory, and all women had the chance to prepare here, and then all women underwent the triasocial stress test. I'd like to show you another study before I come to the data, because you could ask, why not comparing alone against physical contact? Why including the verbal social support? The reason is simple. The reason is that there was this famous study done by Clemens Kirschbaum in Trier in the 90s, and he could show that there's a dramatic difference between men and women. This study is replicated now 5 times. It seems to be a stable effect. So men coming alone to the laboratory show pretty high cortisol stress response. This is area under the curve, so the whole amount of cortisol. Men who are supported for 10 minutes by a woman they never met before reduces stress. And men definitely benefit a lot if they can bring their own wives or partners to the laboratory. If you are female and come alone to the laboratory, this is dependent on menstrual cycle phase, pill, and all these things. But on average, you show much lower cortisol levels as compared to men. If there's a man you never saw before supporting you, this does not really help. The only mistake you can make if you are a woman and go to an application situation, never bring your husband, because you see, this leads to a double increase. And someone from animal research saw that data, and she said, I saw something in the internet that fits to the research that you are doing, and this is this one here. And you can also do this now in humans, and as I showed you before, this is now the black squares. This is cortisol stress responses, women coming alone. Social support by the own husband, again, slightly increases the stress. And we have a very nice stress protective effect if males are not allowed to talk and only administer the neck-shoulder massage. The same picture for heart rate. Yeah, only in the massage condition you have this 30-60 minute long-lasting effect on the cardiovascular response. All these things are, of course, fun to do, but if you talk about treating patients, then physical contact, especially when we're talking about social disorders, is a critical thing. And of course, lactation is another limitation of using the oxytocin system. So at the end, we use, and that's still the only evidence that we have that we can manipulate, at least for a certain time, the levels in the brain by intranasal administration. I show you the first study that have been done in humans. The first study was oxytocin or placebo, double-blind, intranasally, and these were men. Yeah, we're not talking about women, so we, instead of breastfeeding or massage, we administer oxytocin or placebo. And men were asked to bring a good friend or the partner, a person where they think this accompanying person supports me and reduces stress in this preparation period. and the other group was just told to come alone to the laboratory. So we have this interaction between availability of oxytocin in the brain and availability of social support. And then man again went alone to the Trier social stress test. What we see is that we see it not only in cortisol, also in behavioral data, we see the highest stress response in those who had placebo and came alone, and we see the lowest stress response in those who received both protective factors, oxytocin intranasally, and social support. The bad thing you can do is to think that this is a new drug, an anxiolytic drug. That's a mistake some people make when they just have a look on substances. My interpretation was in 2003 and is still another one. The interpretation is like this. If you see that this here is these gray triangles here, this is social support and placebo. That means social support alone is very potent to reduce stress in humans. And what oxytocin does is simply, you could also say, increases this effect that social support conducts. In other words, if your oxytocin system is working and there is the offer of social support in the environment, you can more competently and more effectively reduce stress and anxiety. That's the main thing. So it's a pro-social, more the pro-social thing than just an anxiolytic interpretation. We can do the same study again, and that's what we did. Also man, same design, but this time not intranasal administration. This time we just separated people by their genotype. And as I said, the important thing, one really exciting SNP is this one here. So what you can do is then you separate people in two groups. One group has at least one copy of the G allele of this variant, and the other group has two copies of the A allele. These are 85% of the population, and this is about 15% of the normal population. And what you now see is, I show you the area under the curve bars, which is easier to read. So if you have at least one copy of the G allele, you show this cortisol stress response if you come alone. If you have social support, you clearly reduce your stress. If you are carrier of these two A alleles, you show exactly the same stress response when you come alone. The only difference is when you now have social support, you cannot benefit from this. You would show a slightly but not significant bigger stress response, although you have this famous protective factor. Maybe these subjects for their whole life make the experience that social support is something okay, but obviously not necessary to reduce stress. And of course, we know that these individuals, we are not talking about patients, these are normal individuals. What we know from these individuals is that they also show less empathy, higher stress responses, higher startle reflex, and other. So the whole picture seems that this is something special. You know, it's not a disease, it's not a syndrome, but there's something special when it comes to social interaction. You could also do trust games. That's what people in economics do. I did that together with Ernst Fehr from Zurich. He's someone from empirical economics, and he's interested in trust behavior. And for that, you see that 16 men are sitting in one laboratory, and they play trust games with each other. Very simple thing. 4 games, but you never know with whom you're playing, so it's kind of anonymous. Both players have always the same amount of points. This is now for the cartoon coins, but it's points in the original. Player A can take this and give nothing to player B, which is okay. Everything is paid anonymously later, cash. But player A could also say, I give something, or I give everything, which is not very smart, but you could do it. This is tripled by the computer and goes to player B. Player B can now take everything and leave the laboratory. Some people do that. But you could also be fair and give something back. It's most of the time only 40% what people give, not 50. Question was, when you have higher oxytocin levels in the brain and you are player A, because player A is the interesting one. Player A is the one who has a risk to invest. Would player A change his investment, change his trust in this trust game? And we can say yes. They showed significantly higher trust behavior, and interestingly, the extreme, which was 12 points, the extreme to say I give in all 4 games that I play everything that I have is the case in 45% of the oxytocin group, but only 20% of the placebo group. So there's a willingness, obviously a willingness to accept higher social risks under the influence of oxytocin as compared here to placebo. Another test you probably know from autism research and diagnostics from Simon Baron-Cohen is the reading the mind in the eyes test. And in this test, you have these eye regions and you have to say as quick as possible what of these 4 labels would describe what this person is feeling or thinking right now. And we found in the first study in healthy controls in young men between 20 and 25 that oxytocin increases this ability of mind reading. And this is, of course, not necessary for easy items because these are non-autistic individuals. But if it comes to difficult items where we can also only have a 50% chance to name the right label, oxytocin clearly and significantly increased this ability to understand the emotional information of a face. And this, in this case, of an eye region. So finally, this, of course, is the reason. I just showed you some examples. Is the reason why many individuals who suffer for social phobia, autism, Asperger's syndrome, borderline personality disorder, the parents, the families, doctors, psychotherapists, of course, think about using this mechanism or this substance in treatment. But it's a long way, I think, until we can really say that we have the right to do it. Tom Insel said, director of the NIMH in the United States, we are by nature a highly affiliative species craving social contact. When social experience becomes a source of anxiety rather than a source of comfort, we have lost something fundamental, whatever we call it. And I think for those of you who are working with different kinds of patients, you would agree that if it is something with social, it's definitely harder to treat. You can treat with behavioral therapy quite successfully anxiety patients, but if someone has social phobia, it's a tough job. And that's because, that's the biological perspective, that's because of our way of diagnosis. We have our statistical and diagnostic manuals, ICD-10 and DSM-5 now, and we are still looking for categories. And if you are in the category borderline personality disorder, this is a personality disorder, the former XS2. Social phobia is an XS1 anxiety disorder. Autism is a developmental disorder. And these are really far away from each other within the book, within the manual, right? So from an oxytocin or from a more biological perspective, these are not very far from each other because the social domain is the problem here. And these are only 3 examples. There are many others now in officially registered as clinical trials, but at least social phobia, autism spectrum disorder, and borderline personality disorder are probably the one that come into my mind, at least, when I think about potential oxytocin effects for clinical populations. I would like to give you one brief example for these from more preclinical research, and then at the end come to speculation of clinical use. Do I have time for that? Yes, okay. If you put patients with a severe generalized social phobia in the magnet, first I show you the amygdala responses to social stimuli from healthy controls. Dark green, this is left amygdala, right amygdala. Dark green is placebo, light green is oxytocin. You see, healthy subjects do not really show that much response to a social stimulus, and they just reduce a little bit their amygdala response when they receive oxytocin before. These are the amygdala responses from social anxiety disorder or social phobia patients, which are, as we all know, hyperreactive. They show this hyperreactivity of the amygdala. And when you administer just one single dose of oxytocin before, they show amygdala responses in the range of the healthy controlled subjects. That's, of course, encouraging. It's not therapy, it's just a preclinical study. Oxytocin and autism, there was speculation already since 1998 in studies showing in the plasma that plasma levels of oxytocin are lower in different kinds of autism as compared to control subjects. Today we know that it's quite critical to interpret plasma levels of oxytocin with regard to the brain, but there are other research done in animal models, showing that oxytocin is obviously relevant for approach behavior or avoidance behavior. This is a more recent study I'd like to show you, also using brain imaging, using fMRI, where we show houses or faces to neurotypical control subjects and to individuals with Asperger's syndrome. And what you normally would expect is that the control subjects should show an amygdala response when they see a face and show no response when they see the house. And that's what we find. In the placebo condition, this is the gray bars are the response to the face and the white to the house. And you see that the neurotypical control subjects show this characteristic response, but Asperger's syndrome individuals do not show this response. If you now give one single oxytocin administration intranasally 30 minutes before this experiment, you see the following picture. There's no difference anymore between the response to the faces between Asperger's syndrome and neurotypical individuals. Again, still no therapy. The mind reading test I mentioned, again here, but now done in persons between 12 and 17 years old with Asperger's syndrome and with high-functioning autism. It was a mixed sample here in Sydney, done by Adam Gusteller in Australia, and he found that again, oxytocin increased, especially, of course, for the easy items, increased this ability of mind reading from eye regions in these individuals. Of course, this has something to do with eye contact. It's not a miracle drug that makes people more, the better mind reader. It's simply, if you can stay in the eye region, that's what we saw before, if you can stay in the eye region longer, if you can come back more often to the eye region, you have a better chance to understand what your social environment is thinking, doing, feeling at the moment. And it's hard to socially interact if you do something like this here. And unfortunately, there's no difference between social phobia patients and autistic individuals. They ignore or avoid the eye region. So they miss a lot of social information. And therefore, I like this study also done by Adam Gusteller's group, 2008 already, in high school students, male high school students. If you administer oxytocin or placebo and you just take the eye tracker and ask people, they should simply watch these faces, then you see that under oxytocin, subjects stay longer, come more often back to the eye region. Of course, in the placebo group, also the eye region is most interesting for these healthy subjects, but they also check other region of the face, and there's a significant difference. So it makes easier for you, obviously, if you think about the amygdala control, the cortisol control. Obviously, it's easier for you to stay a little bit longer in the eye region and get more information there. Someone said, to come to the textbooks in psychology or in medicine, you need a cool name. So eye-gazing effect is quite boring, and I'm not sure whether you're familiar with this movie from Humphrey Bogart, Casablanca, you know that movie? And there he's staring for, it looks like hours, but it's only seconds, into Ingrid Bergman's eyes. And he's saying, I'm looking at you, kid. And therefore someone said, we should call it the Bogart effect. And nice idea. Everyone was flying back then after the conference, and then 1 week later, someone sent an email and said it's not a good idea, because Humphrey Bogart was not exclusively interested into the eye region of, at least when Marilyn Monroe was sitting next to him. So it's still called the eye gaze effect. Last experiment I'd like to show you is because there's not only people who think oxytocin is a helpful thing. It's very important that people, of course, experienced psychotherapists tell me it could also be dangerous. Yeah? Think about borderline personality disorders when they get oxytocin and now they don't stop staring at other individuals, even if other individuals are negative in their emotion, that could be a very bad thing. So we did an experiment to find out whether oxytocin makes you kind of blind of the emotional information, that you just make eye contact. And for that we used this test here, which is different videos, and the video always started with a neutral face, and then it slowly changes to a positive stimulus, which is a smiling, happy face, or it slowly changes to a negative stimulus, which is a angry face. No? So you get two different signals, social signals. You get the social approach signal, or you get the social threat signal. And of course it would be bad if oxytocin makes you blind for this very important social information. So what happens when faces are neutral? This is just a replication of work has been done by Adam Guastella. It shows that oxytocin, there's just the black bars here, in the oxytocin group, people again stay much longer in the eye region compared to the placebo group. And then when you slowly realize that the face goes into the positive or negative direction, we see the following picture. Oxytocin, if the face gets slowly happy, still increases. More oxytocin increases the eye gaze, staying in the eye region. And the good thing for therapeutical purposes is that if the face slowly gets angry, then oxytocin does not, does not show higher or cause higher eye contacts than in the placebo group. So that's important because we won't want to have patients that are blind or that are reduced in their ability to read emotional signals from the social environment. Psychobiological therapy is the term that I prefer because I think it's not the same like administering a psychopharmacological treatment in the narrow sense. What we'd like to do is to stimulate a biological system. And it's also clear that oxytocin alone does nothing. If you would sit at home in your kitchen and take some oxytocin, my prediction would be you will have some irritations in the nose, probably, from the conserving material that is in the spray. But I would predict that nothing happens with your behavior and with your cognitions. So it needs a guided change of experience. For that you need a therapist, and for that you need psychotherapy or behavior therapy. And oxytocin can only help a little bit to change this experience that subjects make in these sessions, and then hopefully also transfer this to their real life. We discussed that in some book chapter. I can also, as Chris already said, send you literature if you write an email to me. So we try to at least explain the idea of psychobiological therapy which is more than giving a drug, another drug. Yeah? If you just give oxytocin to all patients that you have that you treat, I think that's a bad example for clinical research and for doing something good. It's more work at the moment for us. It's more work on behavioral therapy and on developing new kinds of psychotherapy for this purpose than just spraying the oxytocin. And I'd like to give you one clinical example because I think this is interesting to see that we are on a good way. This is not from autism, this is from social phobia. You see that there's a group therapy combined with oxytocin or placebo. I show you these groups now. This is not finalized, but I'd like to share the data here with you. If you do this, what you find on the Leibowitz Social Anxiety Scale, which is the important scale for measuring social phobia symptoms, you see that cognitive behavioral therapy, 10 sessions, clearly reduce social phobia, which is good, but the symptoms are reduced more clearly in the group who received oxytocin in addition to CBT. So the combination is better than just cognitive behavioral therapy. That's also giving us hope for other disorders, including a borderline personality disorder. We are doing an inpatient study now for 3 years. It's also, unfortunately, also not finished. We will have data in fall this year, where subjects or patients in this case receive oxytocin 3 times a day for 8 weeks, inpatients in the hospital, combined with dialectic behavioral therapy. So it's again the combination of behavioral therapy, intense kind of therapy, and oxytocin administration. As many other studies worldwide, also running study, so I have to come back in 2 years and hopefully tell you more details. So, summarizing all this, we try to do this also in some more perspective reviews. If you're stressed, if you are in this situation of social anxiety, social stress, we know that this is associated with upregulating all these systems. A good kind of coping behavior is to approach. Females do that more competent and more often than males, and also in humans. But this would stimulate the oxytocin system, and we know that oxytocin system is important for controlling stress and anxiety, something we can call social buffering. And on the other hand, if you stimulate the oxytocin system, nasal spray, but we also have heard that other kinds of other approaches are possible here. Oxytocin stimulates, again, like the trust game, that you show more pro-social behavior. So there's something increasing each other here. And this is only true if everything is okay. If you have problems with your social behavior or showing pro-social behavior, or with your oxytocin system, nobody knows. Well, we can't really say that the oxytocin system is not working in autism, because we cannot measure it right in the brain where we should measure it to come to this result. Then we're talking about different kinds of psychopathology, and behavioral therapy works sometimes, but as I showed you, not so convincingly than in other disorders. So what we're currently doing, and many other labs too, is to combine changing of behavior and cognition with oxytocin administration, something that we call psychobiological therapy. I'd like to thank my group, because they are doing a very passionate job and have a lot of fun, which is important to do this research every day. There are some former lab members who moved on to have their own lab. I showed you data from Francis Chen, for example, Beate Dietzen, and I think also from Robert Kumster. And also a lot of, many collaborators, because we are psychologists in our group. And as you can imagine, for some of these projects, you need other disciplines to move on. And there's a list of collaborators we are working with for many years now. And I also like to show the organizations where the money comes from, always hoping that this is a good thing to get more money from them in the future. And I think that's it, and I thank you for your attention. Thank you.